The overall goal of this method is to prepare nanostructured hybrid particles using carbon nanotubes, or CNTs, and lipid self-assemblies.
This method presents one of the smart applications of carbon nanotubes, where they stabilize nanostructured emulsion of internally self-assembled lipid particles. The main advantage of this technique is that the protocol is fairly simple and it combines elegant properties of both CNTs and lipid nanostructures.
The CNT stabilization produces an oil and water emulsion where the particles have a size of about 1 micron or below, and their core contains a bicontinuous cubic lipid nanostructure. Due to coating of CNTs with biocompatible lipid molecules, their toxicity is anticipated to be low. Recent reports have so far shown that functionalization remarkably reduces cytotoxic effects of CNTs while increasing biocompatibility.
Demonstrating the procedure will be Yogita Patil-Sen, a postdoc from my laboratory, and Amin Sadeghpour, a postdoc from Professor Rappolt's laboratory.
Prepare a 0.2% surfactant solution in water by dissolving 200 milligrams of the surfactant in 100 milliliters of ultra-pure water by stirring it for 20 to 30 minutes. Add 500 milligrams of molten dimodin U, DU, or phytantriol, or PT, to a glass vial. Then, add 9.5 grams of the 0.2% F127 solution.